Good morning, good afternoon, everybody. It is a great pleasure to introduce Professor Paul Strother, a leading figure in the field of micropaleontology, palynology, and particularly in the study of the earliest fossil evidence of life on land. Paul is internationally recognized for his contribution to our understanding of the early evolution of terrestrial ecosystem, with a special focus on the early Paleozoic microfloras. His research has significantly reshaped our view of when and how photosynthetic organisms first colonized the continents, offering new insights into the evolution of the biosphere and the interactions between the life and the environment in the deep geological time. So we are therefore delighted to welcome him today for this seminar organized by the Palynology session of the Micropaleontological Society. And please join me in welcoming Professor Paul Strother. Paul, you can start, and thank you to accept our invitation.
Okay, everyone can hear me now?
Yes.
Yeah, okay. All right, so I really want to talk about the algal plant transition, but based on the Cryptospore record and just a couple of caveats. Talk is really based on my own experience and my own material. I realize other people work in this field. It's not just me, so please don't feel slighted if I don't discuss other people's work. I'm trying to present this story from my own perspective, really, pretty much in terms of, and to do that, I'm going to start. Well, I'm going to try to compose this in terms of three parts. One is that I want to present directly, or in a, I, I want to try to reassure people or explain why I think Cambrian cryptospores uh, uh, are part of the story of plant evolution and the origin of and are part of uh, the evolutionary transition from the algae to the land plants. It's and and just to sort of bear with me, I just want to try to present the rationale behind how I came to the conclusion that these types of cryptospores belong in a discussion about the origin of the land plants. And then the second and third parts are really my view on trying to move away from focusing on when did land plants evolve in terms of a single point in time and try to put it in an evolutionary perspective. Try to look at the evolution of plant development, which is, of course, the definition of evo devo. And, and in I've more recently been trying to move away from just looking at phylogeny and looking more at what fossils can tell us about the evolution of the developmental processes. And part of that has to do with recognizing that there's a huge difference between the, the algae and the land plants. All right, that we think of them as maybe a single branch point in time, but there's a lot of evolution that had to go on to produce and to evolve an embryo and to evolve the developmental processes that produce an actual plant body. And then lastly, if I have time, if I talk fast enough, and sometimes I can talk rapidly, I want to talk a little bit about a new idea I have in terms of the evolution of tracheas, essentially, based on the plant protonema. And so the first section, what is the evidence for showing that the for showing that the Cambrian cryptospores belong to lineages that led to the land plants, and it begins with a story um, in the Grand Canyon. In 1994, I took a camping trip with a colleague of mine from Gettysburg College, an astronomer who wanted to bring a geologist on a trip to the Grand Canyon. So it was totally just camping. It had nothing to do with science. And here we are in '94. We were trying to get to Clear Creek, and we didn't made it, so we camped in the middle of the trail on the way. And you notice in the background at this Sumner Butte location, you see uh, greenish shales in with sandstones. And these reminded me of rocks that I had worked on earlier in Pennsylvania that looked like fluvial sandstones. And it was a surprise to sort of think of these deposits as being near shore, because this section in the Grand Canyon is the classic one for the definition of a transgressive sequence that was described by McKee and Resser in 1945. But it turns out that in the eastern Grand Canyon there is this remnant topology of quartzite islands of the Shinnemu quartzite that stick up, and these islands were overtopped during Cambrian time. So you've got these fossil islands that existed 500 million years ago, that were there, exposed land, when the sandstones and shales of the Tapeats and the Bright Angel Shale overtopped those islands. So here we have one of these islands with this Bright Angel Shale that is abutting right to the edge of the island. And so it's possible to go and sample these locations. Here's another example of how, demonstrating that these are very near to the shore. We've got an example of these boulders that were buried in the Cambrian that are today weathering again. So the debris that's covering this landscape, these Cambrian sandstones with their embedded debris, that same debris is exposed today. So we know that these were islands that were exposed and were dumping their material into a shallow, well, an estuarine ecosystem. And this is also a time of the rise of early rise of animals. This is Middle Cambrian, and bioturbation. Here we have a great example of rippled sandstones that were actively being bioturbated. And what were they eating? Well, here's what they were eating. At least what we see from paleological preparations are what we later became to describe as cryptospores. So these various different spore-like or just globular microfossils, really, when we first look at them. They don't have a particular appearance that looks anything like a land plant spore at all, but I am assured that I can assure you that they are very near shore and probably terrestrially derived. It wasn't 20 years later that I actually put names on some of these things to start to create a taxonomy. And they fall into, well, this taxon Cryptospores. It's very broadly defined, just sort of globular, spore-like microfossils that are encoded or encased in various envelopes. And I've generated a couple of different species, but these are just morphotypes. They don't have a lot of direct correspondence to a genus and species in real life, but at least we have I've started the process of trying to put names on these things. So here are a couple. Here's Spicispora, Omont smooth-walled form that does tend to form, can form tetrads, but also pentads at 1, 2, 3. Well, that's 4, but they're not regularly formed. They're not tetrahedral tetrads. And then Vidalgia, named after Gonzalo Vidal, is another form. It just has a different texture of wall type, but just to indicate that there is some diversity in these forms. So the Cambrian cryptospores, or Cryptospores sensu lato, as I'm going to call them, just a few facts, or from a summary of what we know about these from the Cambrian. They're found only in China and in Laurentia. They begin in series 2, stage 4, down sort of lower middle Cambrian. They occur in estuarine to near shore deposits. They do not co-occur with acanthomorphic acritarchs, so that's another reason to think these are not marine. And there's a nascent taxonomy, a very highly lumped taxa. But there's been a great recent review by Kai Wang, so I recommend, if you really want to read the whole picture of this story, you need to go look up this paper. Review. It's much more comprehensive than what I can give you today. Today I'm just sort of giving you my own story. So why do we think these are relevant? Why do I think they're relevant? Why aren't they just acritarchs, or as Charlie likes to call them, algal scum? and in my view, these are non-marine. They have a sort of a spore-like versus cyst-like morphology. In other words, they don't seem to have cysts. They're not cyst-like in the sense of enclosing a protoplast or having an operculum or opening. So they're not like unicellular algae. They have a laminated wall structure, which I'll show you in a minute, that is just figured out by Willie Taylor. The biology of these is that their meiosis is kind of screwed up. In the simple view, that karyokinesis is decoupled from cytokinesis, and the reason I know that, or we know that, is because when these when these spore-like things divided, they remain attached to each other. So you can you can understand a history of their development and their cell division because they do remain attached, and they remain attached and enclosed. So that's just kind of an accident of biology, but it allows us to know something about how these things actually formed and how they formed with respect to a cell division. They're restricted to Cambrian Ordovician periods, so it's not like the cryptospores are found elsewhere in time. They seem to be unique to this time period. We've looked for them in the Precambrian. I haven't seen them, and obviously when you get into the Devonian and above there, no one's talking about cryptospore polyads from the Carboniferous. All right, so they're not. They are part of an evolutionary phase of essentially extinct organisms. And then lastly, this is where Clinton Foster's work comes in. We've been able to show that there's a morphological continuum with younger forms here. So they are sort of connected in time to the actual bona fide land plant spores. So here's Willie Taylor with Barry Lomax in the Linnaeus's garden in Uppsala, and showing one of the dyad from the Grand Canyon sequence samples. And you'll notice that there are 4 layers, or 4 laminar layers in the gray here, that completely enclose 2 dyads. I've done false coloring here, but this is the lumen and the sort of folded over spore body that then has a single thick layer with these sort of this distinctive scabrate form. So we can tell, for example, in this way, we can tell the sequence of wall formation in this because of the common wall that encloses both of the dyads. So here's some of Willie Taylor's work. And long and short, the laminar nature of these Cambrian forms is very similar, actually, to the laminar nature of basal laminae in certain liverwort embryophytes living today. This is a Gammachetes, a form from the later part of the Cambrian in Wisconsin. The cheese state, hence the name Caesarius. And Willie's made 250 slices through this particular specimen that you see here in the middle. And on the right, we see the topology of that selected specimen, which is a little strange. It looks sort of like we have a packet, and a packet of 2 of the packets have 2 dyads, and 2 of the packets have 4 sets of 4 dyads in them. This is one of my favorite specimens. It comes from Tennessee. I call this specimen "This Will Change the Way You Think." So we show an outer wall, which presumably was the spore site, and inside that spore site is a tetrad and a dyad. And that makes me think, or allows us to, as I said before, because these occur together and they have a topology with them, we can make predictions, or we can reconstruct the division patterns that would form such a thing. This is a very confusing diagram, but the point of it is that cytokinesis, that is cell wall formation, is not coordinated directly with karyokinesis. In other words, every time the nuclei divide, you don't immediately form a cell wall or a cell membrane plus a cell wall around those things. So they're decoupled in time. So that allows us to, that's the model for an example of how we can have a tetrad and a dyad inside a generative cell, a single generative cell. And it's interesting, this is the work of of David Haig at Harvard. He has a number of papers describing the nature of cell division in the life cycles of various streptophytes, including Coleochaetae, which until a couple of years ago was considered the closest relative to the land plants. And basically, these here's some older data from Hopkins and McBride in the 70s looking at Coleochaetae. Basically, these the cells in the zygotes undergo multiple rounds of nuclear reproduction, essentially karyokinesis, without forming cell walls. And so, at the end of the day, the number of zoospores that is formed from a single diploid zygote ranges from somewhere between 2 and 32 or something like that. And this was all described by Linda Graham back in the early 90s. So here's kind of the summary of what we know about the Cambrian cryptospores, basically, and their relationship to modern organisms, the modern algae, essentially the charophytic algae. We know something about development in terms of how wall polymers are formed, or the topology of wall polymers. And we know that karyokinesis and cytokinesis are not tightly coupled. It's interesting in the, I think that'll be the next slide, is the study of sporogenesis in bryophytes tells us something about the bryophytes' spore formation kind of predicts where the cell cytokinesis is going to take place. I'll come back to that now. Actually, I need to talk now about the next phase, about completing the our sort of stratigraphic occurrence of cryptospores in general. So seemingly different topic, but I'm going to work from the younger to older now, and so we know that we have plant axes. This is from the late 60s. Plant axes in the Wenlock, and Amphibisporides as the trilete spore, of course, is a marker for embryophytes. There's always been a slight gap with the triletes being a little bit older, occurring in the Clondovere, and the first axes occurring a little bit later in the Wenlock. But that makes sense. Spores are more widely dispersed, and we expect to see them in the fossil record. And then starting in the early 70s with Gray and Bucco recognizing permanent tetrads from the Lower Silurian Medina group in upstate New York, and basically relating these to certain permanent tetrads that occur in the liverworts and in hornworts. This was followed very well a few years later by a study by Pratt et al. From Virginia. They essentially had laterally equivalent units in the Massanutten sandstone in Virginia, and they have found the same thing that Gray and Bucco talked about, except that they also found tubes and dyads as well, which Gray and Bucco at the time didn't talk about. And then Strother and Travis in 1979 began the taxonomy of these things by describing this guy here, the Tetrahedroletes medadensis. And this was based on, just a personal note, this was based on originally on a class project that I had with Al Travers at Penn State from 1973. So anyway, it took 6 years to publish the paper, but I ended up going to grad school in the interim. But at the time, the Gray and Bucco paper was the only one, and so they were definitely first to recognize this. And then it wasn't until 1984, another 5 years later, that John Richardson at the Natural History Museum introduced the term cryptospore for these tetrads and dyads that were permanently fixed together and had not broken apart as separate individual triletes. So here's an example of material that my graduate student John Beck worked on at Boston University many years ago now, 20 years ago to be precise. Actually, these were not that well preserved. These ones from Allenport, I think these are actually IR images of the specimens. But anyway, you begin to see there's tetrads and dyads and various other monad forms. Here's an example from the original deposits where Tetrahedroletes was described from the Tuscarora Formation in central Pennsylvania. Here's some unpublished material from Ontario that's recovered right at the Cambrian, I mean, sorry, the Ordovician-Silurian boundary. Anyway, more tetrahads, tetrahedralides types. And then in the late 80s, in the actually mid 80s, Milada Vartova in Prague started publishing published material from the Prague Basin. She's more well known perhaps for her acritarch work, but amongst those acritarchs in the Kosovo Formation, which is late Ordovician, she found quite an abundance of various cryptospore, what we would now call cryptospore morphotypes. And then in the 90s, Charlie Wellman began in his PhD thesis working on the keratic and looking through the type sections and then finding lots of different kinds of dyads and also generating an additional number of genera to the overall taxonomy of these forms. And finally, working on some material that came from Saeed Al-Hajri's master's thesis at Penn State, we described these early cryptospore assemblages from Saudi Arabia, from the Darwinian, and that's kind of retained itself or maintained itself as a baseline of the beginnings of tetrahedral types of tetrads and different wall types, et cetera, et cetera. So in the Hanader member, there are good examples of tetrahedra grayi, smaller tetrads, and then also Rimosa tetras, which is less tightly bound than Cryptotetras and tetrahedralides, and then also dyads. The dyads go along with these assemblages. And in my view, then these are really the Darwinian forms are kind of the beginning of the forms that really look like they belong to embryophyte spores. As of 2018, not that long ago, if you look at a composite sketch of what I just described, we had sort of a gap between the Cambrian forms, which were much different from the trilete forms. And on the left-hand side of the diagram, I've just taken pictures from various published forms, and they're all to the same scale. So you kind of notice right away that the cryptospores that from the Cambrian are actually kind of smaller and much more irregular than the forms that begin in the Darwinian and then proceed up to the point where we get Cooksonia coming in the Wenlock. And then here's the story about Clinton. I like to include these a little bit, these kinds of stories about serendipity in science, I suppose, in the same way that the Cambrian cryptospores came about accidentally just because I happened to be camping. I wasn't looking for this on purpose, and the same thing. I was at a Lyle meeting at the JOLSOC in 2019, and Clinton showed up. I don't know if he had some business or some trip, the reason to go to England, but he just came to this meeting and then told me about these slides that he had from the lower Ordovician, and would I mind taking a look at them? And I said, yeah, sure, send them. So when I got home, he sent me these things. A box of slides from the Nam-Meat Formation, which is actually, well, it's Tremodockian. It's the lowest unit of the Ordovician. Anyway, it comes from an old Clinton, I think, in his quote-unquote retirement, has been going through looking at old cores and finding all kinds of interesting things from Australia. But this material from the Nam-Meat Formation is actually quite interesting. We know from the work that Taylor has done that these thickenings and sort of folds in the walls actually are indicative of multilaminate wall structure. So that's kind of one thing that tells me that these are related to cryptospores. But we also found planar tetrads, which if they had been found in the Cadian, we would have just thrown them into tetraplanari sporites. I mean, they would just be formed, they would be regular cryptospore sensu lato forms. And the same way with open tetrads. Here's an example of Rimosa tetras, which again is found in the Darwinian. I've already shown you a picture of that from the Hanader material. And then in the dyad forms, there are paired dyads which are very similar to Dinosporus geminus, which is this Cambrian planar dyad form, but also one that we included in the plate in this in our publication. Um, this is a specimen of Dyadispora Muris attenuata, which is from the type slide of Dyadispora from central Pennsylvania in the Silurian. And there's not a lot of difference between these forms and these lowermost Ordovician forms. At least I don't see that much difference between them. And so the interesting thing about Clinton's material is that really helped us create a morphological link and a continuum from Cambrian forms all the way up into our our Ordovician and eventually lower Silurian forms. This is a compilation that we did. This is Willie Taylor's work compiling occurrences of Cryptospores. It's an incomplete data set. If you want to see the complete data set, you need to go to Kai Wang's paper that was just published. They have much more complete data here, but this was our version in 2024. Just compiling from the literature, from our own material plus the literature, and it's quite incomplete, but it does show you a temporal distribution of these different forms. And that's kind of the end of the story in terms of, I mean, we're missing things. For example, we don't have anything from the Phloem within the Ordovician, and I suppose as time goes on, I would assume that we would find more terrestrial-derived materials from different parts of the world, and these are derived, of course, from all the way from Russia to Baltica, Laurentia, and China, and now Australia as well. So they are worldwide in terms of Ordovician to Silurian, although the Cambrian forms, again, are just found in Laurentia and China. I now want to move to, that was the 15 minutes of the talk, which is actually half an hour, so I'm only half as fast as I need to be. Which is, how did the algae evolve complex multicellular, instead of the question, when did land plants evolve? And I try to move on to why that to me is more interesting than just the timing relations. And we tend in paleontology these days, particularly when we're trying to incorporate molecular data, to be concerned with phylogeny, which is really a focus on the pattern of species relationships. And so we know that the land plants came from the charophytes, and the zygnematophytes are the closest, our sister group. That is, genetically, they're closest to the land plants. But it's kind of absurd because Zygnema is just a filamentous alga, and it doesn't even have flagellated cells in its reproduction, blah, blah, blah. It's morphologically miles away from Cooksonia. And so we really want to be looking at not just these branching relationships of species, but we really want to focus on the evolution of development, the evo-devo aspect of evolution, which in this case, for the closest sister, for the sister group, involves gene loss and genomic assembly. If we put this on a timeline, looking at the last common ancestor, we must, this is not my data, but there's data of gene loss in Zygnema. Oh, the image up there is a zygospore of Zygnema. And then genomic assembly in terms of piecing together what we see in the fossil record. We really don't see a lot of speciation of course, we don't see speciation of embryophytes until after the embryophytes evolve. But once we have speciation within the embryophytes, then looking at phylogeny and species relations is fine. But it doesn't work in this Cambro-Ordovician time period. So we need to go and look at what the biologists think of how the algae turned into land plants, and then lastly, what relevance is the fossil record to that question? To what extent can the fossil record help us or impede us in that understanding? And this begins with Bauer in 1908, and then I'll discuss a little bit about this issue of bryophyte sporogenesis, and then not dwelling on the spore-pollen transfer hypothesis, but all these elements are related to and can be related to the fossil record of cryptospores. We begin with Bauer, again in 1908. And Bauer's idea, or his antithetic or interpolational hypothesis, I think he preferred the latter term, because the idea was that we started out with a plant life cycle or an algal life cycle and then interpolated the sporophytic phase. Um, uh, and his idea was that, well, the sporophytes evolved in response to terrestrial selection and that the spores evolved first. This is a long uh, quote from him, um, but they basically, the sporophyte formed, in his theory, through a delay of meiosis from sporogenous material, or sporogenous tissues. And we'll look at that. Here's my modern version of his interpolational theory, starting at the bottom. The life cycles in blue are considered to be essentially aquatic aspects of the life cycle, and the green are supposed to be things that happen in response to growing on land. And then I've added, just earlier this morning, added the little sporopollenin issue to show that we start out with an organism like Coleochaete that has a zygote with a sporopollenous wall, as described by Delwich and Linda Graham back in the 1980s. And then that sporopollenin is transferred from the zygote or the 2N wall to the 1N wall of the spores. That's the nature of the sporopollenin transfer hypothesis. But we begin this genomic assembly within the life cycle at the algal stage, and we end with the evolution of complex multicellular development that is incorporated into the into an embryo, but into the genome of a zygote that produces the plant embryo. So do we have evidence for his theory? And there are three aspects of evidence that exist for direct evidence. One is looking at extant bryophytes, and then the second is sort of obvious, is the fossil spores occur before the actual plants. And there's a little side story to this that I'll focus on about these spore-producing thalli that was predicted by Bauer, and we've actually found them in the lower Ordovician. So here's from Brown and Lemon, 2011. This is a copy of their diagram showing about their observations on spore development. One is that these are heterochrony, basically shifts in timing that give results from an algal ancestor to the actual bryophyte system, which is on the bottom. And that includes the idea that the sporophyte, that the zygote wall, the sporopollenous wall on the zygote has been delayed, and so eventually the wall is applied to the individual spore types themselves. And then the other thing is that the bryophytes predict the positioning of where cytokinesis is going to take place by these pre-prophase bands and then quadrilobing, none of which exists in the algal precursors. So this is a little bit of story about the so-called spore thallus, this Grododowan. These we see in the Cambrian in some cases clearly aligned packets of what look like spore tetrads, or cryptospore tetrads. And we also found this with Clinton in the Nambeat microflora. It's two-dimensional packets of spores that follow very geometrically regular patterning. And we found this with working with Marco Beckley in a place called Fossil Mountain in Utah. I love the name of it, Fossil Mountain. And it's kind of it transgresses. It goes across the Dapinging and Darwinian boundary in the Middle Ordovician. Here's some examples of what we see in these Grododowan forms, where you see spore packets. They're clearly geometrically regular and showing regular types of divisions. Here's another example. In this example, you can see that the reproduction is not perfect. So here's like an extra division that's occurred here that produces two extra packets, and then again one in the back here. So they're not perfectly aligned, but they're very closely regulated, geometrically regulated in terms of the cell divisions that have produced these packets. Here's an example of a rosette form, a sort of a rounded form, and this is very similar. You can follow the trace of cell divisions that must have taken place. Again, the topology of the specimen preserves a sequence of cell divisions, and those divisions match very closely what we see with living Coleochaetae. But at the end of the day, it's important to realize that these are not they're not vegetative in the sense that at the end of the day, these sheets of what look like vegetative cells, vegetative tissue, are actually producing spores, and they're actually producing diaspores of dyads, of tiny little dyads. So they're spore-like cells that form a thallus. So now, sort of, I want to go back and repeat that sort of summary, um, looking at the spore record again that was completed by or assembled by with the help of Willie Taylor in terms of categories over time, and now I want to add in a little bit of stuff that seems well, it's kind of at the very end of this story. That is cuticles and tubes that occur throughout the Silurian, actually. If we look at the most modern sort of the molecular biological view of this story, coming from John Bowman, this is his Nature Plants Nature Plants paper from 2022. This is kind of like what I did during COVID, sort of, where we look at the various molecular components of the idea that over time these algal starting with chlorophyte, then charophytes, then streptophytes, and lineages acquired the so-called genetic toolkit that eventually was incorporated into the land plants. And so these include transcription factors, signaling molecules and pathways, novel biochemistries, so to speak, and then other key innovations in what we would describe normally as characters that are found. And some of those characters are capable of being seen in the fossil record. So sporopollenin, cutin, and other resistant biocompounds, polymerized compounds, have the potential to be seen in the fossil record. And here is Bauer's I'm trying to get back to his diagram, a composite diagram where putting on a timeline and looking at the whole picture. So here's the cryptospore record that's sitting on top of what we know about a little bit about fossil bryophyte type organisms, the vascular and provascular lineages, and then the modern lineages or the extant forms in terms of their branching and their distribution in terms of phylogeny. But what we see at the bottom, where we are trying to form an embryo, is the acquisition of the toolkit that takes place over this period of time. Here's my simplified view of that same diagram without all of the botanical stuff. But the point is that we begin this genomic assembly back in the Cambrian, and what we see at this boundary point in the middle Ordovician is the beginning of a bryophytic meiosis. In other words, the regularization of the production of spores in the life cycle. We don't see the production of an axial sporophyte at this time. So where did that axial sporophyte come from? And this is part 3 of this talk, the idea that perhaps the protonemal phase of bryophytes before they completely evolved, sort of in the last phases of genomic assembly. Now, if you know something about the life cycle of mosses, they produce this filamentous phase in the life cycle. When the spores germinate, the filaments come out, and they produce a structure called a protonema. That's a mass of growing filaments, and the protonema will overwinter by producing a brownish-colored thickening exopolymeric wall material that produces a structure called a colonema. And my idea is that, well, maybe those tubes that we see in the Silurian latest Ordovician and Silurian record, uh, could they possibly be related to some kind of a form that originated as a protonemal cell type of tissue? We see some examples of these in the Cambrian. Well, we're kind of like, they look sort of like moss protonema. They've been published but not emphasized, I would say. And I want to show you just some examples of what some of these banded tubes look like. They've been worked on by other people, described by many people back in the 80s and into the early 2000s. There's not a lot of my work, really. The prevailing arguments have been that they belong to the fungi. I won't go into this too deeply, but there is some more recent chemical evidence that indicates that maybe they're not really fungi based on chemistry. And other than being tubes, they don't really have a lot of synapomorphies with the fungi. Here's some examples of Cambrian forms, and the Cambrian forms actually these wefts of thinner tubes, and this is an example showing on the right-hand side the extant colonemal cells of mosses, and in the middle there are some examples of slanted and weird cross walls that are characteristic of the cellular aspect of these forms. So here's the idea that the charophyte algae evolved the capability of growing elongate cells, and that these led to variations in their wall construction, and that the genetics that produced these types of cells was later incorporated as kind of proto-tracheary elements in an actual embryophytic plant type. Anyway, that's not a very good explanation, but that's the idea. So here's some examples put on a timeline. We see these kinds of things all the way back to these are from the lower Silurian, but they go back to the uppermost Ordovician. So homogeneous type wall types with lots of fracturing in there that are showing there's some brittle. And then in G, there up on the right, you see some forms that look more like fibers with tapered ends. One of the things about the banded tubes is kind of interesting that they are typically broken and don't have clearly defined terminal ends to them, but this example in C appears to be an end, and also in G having complete ends to them. So the preservation of these things is pretty good in New York State and in Pennsylvania in the Silurian. So here's some example of walls that have a fibrillar construction to them. And then here's examples of walls that are homogeneous on the outside but have annular thickenings, completely different types, perhaps. But there's variation in the nature of the spiral thickening. So here in C, E, and F, you're going from, B is that so you have one spiral, a single thickening that's spiraling, and in C we have two that are spiraling, also in E, pairs of spirals, and then in F there are actually a series of 5 different spirals that are forming helical structure. And then when we get to G, we have an entirely sort of fibrillar nature of these thickenings that are occurring. So what do we have? So they do seem to show some level of increasing complexity from the Hernandian to the Wenlock. And so that there appears to be some morphology that is showing an increase in, again, an increase in wall complexity. But the middle lamella, the next step that we don't see in the fossil record is the fusing of forms, a fusing of tubes into essentially a woody tissue where they're glued together. And so you need to evolve a middle lamella. And so my argument would be that the evolution of the middle lamella, that is the apoplast that's gluing tubes together, is what was the evolutionary novelty that occurred in the embryophytes that made the axial structure of the upright sporophyte. So here's our summary of a timeline of what I think is going on with these protonemal and pre-tracheary elemental forms, going from the Cambrian observation of just sort of simple protonema that they existed back in the Cambrian, and then when we get to the latest Ordovician and on into the Silurian, we can see this progression of increasing complexity that was occurring. And what's interesting about these in terms of genomic assembly is that we can now look at the nature of the materials that are needed to produce those types of structures and think of those as essentially markers. That is, the fossils become markers of biosynthetic pathways that and as time points, tying those the origin of those biosynthetic pathways and putting them into a timeline. Let's see. Yeah. So again, here's another example of that timeline of sort of adding things together and trying to fit all this into a timeline that fits Bauer's antithetic model. We start out with cryptospores per se, and then we end up with these spore thalli, two-dimensional spore thalli, but they're all done in the context of this, what I call endosporic reduction division, as a way of describing the charophytic production or charophytic cell meiosis, the cell cytokinesis that's occurring within before the establishment of normal embryophytic meiosis, where we get a very morphologically constrained topology of tetrahedral tetrads. So eutetrads or tetrahedral tetrads come in somewhere in the lower to middle Ordovician. The sporangia that are described by Wellman et al. From 2003 come in sort of in the middle to later part of the Ordovician. And then finally, when we get to the Silurian, we have true triletes and cuticles in the latest Ordovician, but they're not very common back then. But eventually, in the Silurian, we see them tubes, banded tubes, and then finally true tracheids and embryos. So here's getting to the end here in terms of thinking what we're really talking about here. The origin of land plants. Two aspects of this. One is the process of the origin of land plants, which is the evolution of complex multicellularity as a biological process. And then the result of that, in terms of timing, which has to do with the evolution of the terrestrial landscape, weathering the Kármán cycle, et cetera, et cetera. And the timing of that process from the basis of molecular data is actually overlapping with the Cambrian explosion and the GOBE. These are the events that are described for us by our invertebrate colleagues and paleobiological colleagues. But the process shows that spores evolve first, and that a series of co-opted algal genes plus de novo plant genes are what was incorporated, or what were incorporated into the earliest land plants. And the question of molecular clocks, I've sort of mentioned this, only briefly, and it hasn't really been challenged that much yet. And that is it possible that the reason the molecular clocks are telling us that land plants are so much older is that somehow they're tracking older DNA that's been repurposed in the land plants, and rather than the actual plants themselves. So everybody wants to know when the embryophyte's involved, what's the time of that? And my argument right now is that if you just read the fossil record the way we have it, what we see in the fossil record is that plant axes, that the sporophytic axes, still don't occur until the Wenlockian, based on Diane's material and also material from the Prague Basin. The trilete spores come in earlier, certainly in the latest part of the Ordovician, but that what we think of as the conventional origin of land plants from the Darwinian is really just the origin of meiosis in plant sporogenesis, is the canalization of the meiotic process. And that probably happened in some plant-like organism prior to having a sporangium that was sitting on top of an axial stalk. That is it for me. So I need to find where everybody else is in the Zoom meeting right now.
Thank you, Paul.
I can't see anybody, so that's why I need, I need to find those. I need to find you guys.
Mm, we can stop sharing, maybe.
Questions?
And I'm trying to present their layout. Sorry, I'm so technologically confused here. My screen is doubling itself. Okay, I'm going to stop sharing and then we'll.
Ah, okay. Now
Oh,
you can.
now I can see everybody.
Okay.
So, all right, shoot.
Questions?
Yeah, there is one in the chat. Sheila, do you wanna say it aloud, or you prefer us to read it? You can unmute yourself. I think we can read it. Do you have the chat?
Yeah. There is a question. The Cambrian is generally shallow marine, at least in Morocco. How to explain the existence of charophyte? Have you established any correlations with trilobites?
Um, yeah, I've had that question before. Most of my experience in the field has been with the Grand Canyon. Um, and in the Grand Canyon, there are trilobites in these beds, but in the upper part of the Bright Angel Shale. But the trilobites are always associated with storm beds that include echinoderms too, and apparently these are beds that have um, uh, moved shoreward from a more open marine setting into the embayments or into the estuaries. Um, and they all occur within, well, it's the Glossopleura biozone that lasts, I don't know, it's like 2 million years or something. It's a pretty short time interval. And it's also co-occurs, it's the same time interval as the Burgess Shale, coincidentally, further up in Canada. So if you look at the, the, it turns out that there are some trilobites that are capable of getting very close to like crawling onto land. You have to look at the people that have done this are Gabriella and what's her husband's name? Um, Luis in Saskatchewan, the Argentinian couple. They really are the experts on this, and they've actually shown that in some cases the trilobites can exist in these estuarine and uh, freshwater affected deposits. I don't know that's anything about Morocco. Sorry.
And sorry, I have a question. Um, Very short. How can the terrestrialization during the early Paleozoic can contribute to the deoxygenation that became in that period? Surely there is a link,
Yeah.
okay?
So I think that if you look at I mean, this is, I'm not an expert on this, but if you look at the basic model, that is the geocarb sulf model from um, what's his name, Berner and colleagues, um, all of those models show high levels of CO2 in the Cambrian and then dropping through the Cambrian and sort of bottoming out actually near the end of the Devonian. Um, so, and that with little humps in there. Um, and I've always thought, well, that means that basically, in order for the oxygen to drop, you need to bury carbon. And you don't really see that carbon burial until the Devonian and the Carboniferous. Um, I mean, that's just the basic aspect of photosynthesis. That if the carbon is not um, uh, respired back into its gaseous form of carbon dioxide and oxygen, so you need to bury carbon to have that happen. So that sort of implies, and part of that buried carbon then is what I showed in the very first part of the talk when I said, what are they eating? There is a pathological record, and this has now been expanded actually by Butterfield and Tom Harvey in particular, on the looking at the small carbonaceous microfossil, looking at the pathological component of essentially buried carbon. I mean, it's buried carbon that's animal carbon, not plant carbon, but so my guess is that began that the associated with the accumulation of algal mats or microbial mats surfaces during the Cambrian, and then the burial of carbon from those deposits is what began to bring down that oxygen. I'm sorry, to increase the oxygen and bring down the CO2. So it
Okay.
began in the Cambrian near the
Okay,
Cambrian
yeah.
boundary, but it didn't really it became significant in terms of carbon burial and oxygen increase in the in the Carboniferous, right? I don't know if that answers your question. I don't,
No, no, no. Okay, yeah. Okay, yeah.
but it is a little bit weird that, in other words, that you might expect that we would need to create, once you create trees and forests and secondary wood, and that wood gets buried, that's what causes the transformation of in the oxygen and carbon dioxide story, and gives us our modern balance of having 20% oxygen and 270 parts per million CO2 probably occurred in the Carboniferous first. But
Okay.
prior to that, would have been lots and lots of CO2. It would have been warm as heck back then, in spite of the Ordovician glaciation.
Yeah, the terrestrialization should contribute to the development of soil
Right.
and the weathering too. There is a signal during in the Ordovician,
Okay. There is a what?
a signal because I think, I guess, the terrestrialization should contribute, the colonization of land plants should contribute to the development, increase of soil, the formation, and the weathering too.
Yeah.
Is it so?
Well, I mean, that's a really interesting question too. Kennedy and Nathan Kennedy in 2009, I think, or 2005, had a paper called on this very question, associated with different clay formations, the weathering of different kinds of clays, the smectitic clays versus illitic clays, began, I think, around 600 million years ago or something like that, into where they began
Okay.
to transition, and that's totally microbial in nature, which is still what we're talking about with regard to the cryptospores. Essentially, they're these pre land plant charophytes. I mean, they're effectively part of a microbial ecosystem, not upright plants with roots and things like that. But it's certainly all of these things. Again, going back to the into the maybe even the mat grazers of the Ediacaran, they are producing organic acids in mat surfaces. Um, so those organic acids are what are going to be attacking feldspars and causing a pH, lowering a pH in soil type environments, soil like environments, I suppose. Yeah, so I would expect to see that starting, at least from that Nathan Kennedy data, starting around 600 million years ago. And that is so that in terms of terrestrialization, there's a microbial component again that begins earlier on, and then something happened in the Cambrian, as far as we know, with the development of these cryptospore bearing organisms. We don't know what they look like. I mean, I think they might have maybe just sort of thalloid, but I don't really have any really substantially cellular based evidence of macroscopic form uh, that was producing these things. Well, maybe Kai has there's some things like rosettes of spores with some tissues around them that he's seen in the Cambrian as well, but we don't have a lot of information about what these plant-like things look like, other than the cryptospores.
Other questions? Okay. Thank you, Paul.
Well, thank you
Thank
all for
you,
listening.
everybody. See you in Munster, probably. I hope.
Yeah, and
Okay.
Clinton,
I just
enjoy your sleep.
Yeah.
I just want to mention was last thing because of the how well received this palynology series have been, the organizers, we have decided to do a second year, so you all will soon hear about it, and we start thinking of potential guests. So if you have any ideas, please send us an email. Okay. Thank you, everyone.
Ciao.
Thank you.
Ciao, grazie. Ciao Paul. Ciao Clinton. Ciao. Ciao Natalia. Ciao ciao.
Au revoir.